Jai Hind everyone. I am Pankaj Kumar. Today's lecture is about what? It is about the amino acid and all. So in amino acid, we'll see the following things as we mentioned here. The following things what we are going to read. The amino acid isoelectric point, Henderson-Hasselbalch equation, problems and equation related to these things we are going to read. So if you'll see the important things about the protein, it can be like present everywhere and it is very fundamental important things for the life. So either it is an enzyme, or either it is your nails, either it is your bones, either it is your cell matrix, either it is your vision, either it is your listening, speaking abilities. These all because of the lights. As you can see, the fireflies which have the luciferin, which will produce the light during night and days as well. So the lights will be produced because of the protein only. Or if you'll see the horn, seals, and the teeth, all these things structure is because of the protein. So protein is very important as it is helping in the transport of the gases via the RBC. In RBC, what is there? Hemoglobin, by the help of hemoglobin. So these are the different important things in which the protein is involved. First, amino acid was discovered, that is a asparagine. It is asparagine. So in this asparagine, if you'll see, asparagine is first discovered, and it is discovered by the Lewis and Perrier. Lewis and Perrier discovered from the asparagus. From where it is discovered? It is discovered from the asparagus. So mostly the earlier the name which we discovered, it is related name we given. Like if it is discovered from the asparagus, we given the asparagine. If you'll see the another amino acid like a glutamate, glutamate is discovered from the gluten, wheat gluten. Okay, wheat gluten. And then if you'll see the tyrosine, tyrosine is discovered from the cheese. Tyrosine is discovered from the cheese. And glycine, if you'll see, glycine is name given because it is a sweet. So glycose is a Greek word in which what is the meaning of glycose? In a Greek word, it is a sweet, and from that, because from that the glycine we use the word glycine. What does it mean? Sweet. Maybe this amino acid is the sweet in nature. Okay. So let's see here. Let's see here. The basic structure is what? Alpha carbon will be there, and this carbon have the how many bonds? The four valencies will be there. So in one side you will find the CO minus will be there. Another side S3N plus will be there. One side you will give one H and another is R. So this is the called as a alpha carbon, or it can be called as a chiral carbon. Okay. So why it is called as a chiral? If you will find in place of this functional group, this is what? This is a functional group, and in place of functional group, if you'll choose the H as well as, okay, if you'll choose as the H, then there will be no mirror image will be looks. There is a like same mirror image will be there, and then it cannot form a isomers. Okay. Now, if it will be CH3, then you will find there is a change in the isomer, and that mirror image will brings the non-superposable image. Then it will form a isomer. In that case, the amino acid will called as a optically active. I will try to show you here in coming videos also. Don't worry about it. So except write it here, except the glycine, except glycine, amino acids are most amino acid are Optically active. Optically active, or you can say it have a chiral center. Active means what? Optically active means what? The chiral carbon will be there. Optically active. If I am saying optically active, it means it will form the isomer also. Okay, it will form the stereoisomer. Okay, isomer. So you can find the relation between the chiral. If it is a chiral, means there is a different groups are attached in their valences. It is not same. And if it is a optically if it is a chiral, then it will be optically active. Means what is the meaning of optically active? The superposible image will be non-superposible image will be formed, and then it will give you the stereoisomer. Except the glycine. Note it down. Except the glycine. Now, if you'll see the amino acid, there will be around 300 amino acid in your till now we know. Out of this 300 amino acid, only some amino acid which will help in the protein synthesis, which will present in the protein synthesis, and these are only, if you are saying 20, then add 2 more. Okay, 20 plus 2, that is a 22 amino acid will be there till now, which will help in the protein synthesis, and remaining will be like a um, remaining will be not synthesizing a protein, but they have an important role, and that's why it is called non-standard. So we divided the amino acid into two parts. Protein synthesizing will called as a standard amino acid. What it is called? These are the standard amino acid. Okay, and some are the non-standard amino acids. So you can minus it. 300 minus 22, it will be 78. 378 amino acid will be non-standard amino acid. So hope so you understood these small things. Now we are continuing with our lecture, in which we will see the amino acids, the naming of the amino acid. What we'll see? The naming of the amino acid. Naming of amino acid have a, either you got the 3 letters code. Okay, there will be 3 letters code for the amino acid, or either it will be only 1 letter code. So the 1 letter code is very important, because it is important to store our data in a very small space. Okay, so it is in like digital data is a very high much, and we cannot store the all the data in form of 3 letter or the like a amino acid name. So what we did, we changed the 1 letter amino acid. So this 1 letter amino acid, amino 1 letter naming was given by the Oakley day off. Oakley day off. And she is the only one who pioneered the bioinformatics, who pioneered the bioinformatics. So the first letter of the amino acid is unique for the amino acid. That's why given the following name. Now you can see. Sorry. So as we know, as we seen that naming of the amino acid is given by the Mokle Day of. She is the pioneer of the bioinformatics as well as. So the way of giving the naming for the amino acid are the following. Sometimes the amino acid are unique for the for their name. So we give the name 1 letter name by their symbol only, first letter, like cytosine, histidine, isoleucine, methionine, serine, and valine. So these are the naming. Now if you'll see, the first letter which is not unique but it is assigned to the amino acid which are more common in the protein. Means if I will see the leucine and lysine, so leucine are more frequently come in the protein but lysine is not very much frequently come in the lysine. So that is called, what it is called, this is called according to that they gave the name AGLPT. So AGLPT is what? AGLPT is the alanine, glycine, leucine, proline, and then threonine. Okay, now if you see the phenotypically suggestive names. So now there is a another amino acid. If I will tell you, mm, arginine. Arginine also start with the A, and alanine also start with the, and arginine also start with the A. So we cannot give A to the both the symbols. Already we give the A for the alanine. Now this A cannot given to the arginine. So what we'll say, while speaking you will get the R first. Okay, arginine. So we'll give the arginine R, phenylalanine F, phenylalanine for F, Y for the tyrosine, and W for the tryptophan. So our maybe in our Indian English, the W and the Y will not come in the tyrosine. Maybe in the another accent it can be. So they given as the Y and W, Y for the tyrosine, W for the tryptophan. Now the rest are suggested by their name. It is like difficult to say any amino acid with the D. But if you'll see aspartic acid. So dick means what? D. Aspartic acid will be D. And then N will be what? N is the asparginine. Asparginine means N. E is for what? E is for the glutamine. Okay, E is for the glutamic acid. Remember it. E is for the glutamic and Q is for the glutamine. So I remember this Q and E there is a little bit different, like confusing. So I remembered Q will be the queen and queen means glutamine. Queen and glutamine. So remember it, glutamine. So glutamine is the queen. Okay. So like that you can remember for you also. Now, amino acid are classified according to the R group. If you see the R group are in the different forms. Like either it will be hydrogen, either it will be aliphatic group, either it will be any aromatic group, either it will be anything. Okay, we'll see one by one. So, non-polar, if the R group is there, which is non-polar, my video is paused. It's fine. I'm going to hide it now. Still, it is like this only. Okay. Now, now you see the non-polar aliphatic group is R. Aliphatic group means what? Like hydrocarbons chain will be there. Okay. So that will be not going to give any charges. Like if you will see in the at pH 7 there will be no charge in it. Neutral. So at pH 7 non-polar are what? Non-polar are the neutral in nature. Non-polar are neutral at pH 7. Okay. So this is the glycine and we already seen glycine will not able to give you the enantiomer because it is optically inactive and it is not going to give you the any stereoisomer as we already said. Okay, but if you see this alanine, proline, valine and any other, the R group, these all four valencies are different. These all four valencies are different in this. So in one side if you will see there is a carboxyl group, another side amino group and another side H, and one side it is a methyl group. So in this case alanine, there is a complete different valencies, different groups in their carbon valencies. So it is a optically active, and if it is a optically active, it will give at least two stereoisomer. It will give two stereoisomer. It is non-superposable image it will give, so it will called as a enantiomer. Enantiomer. So enantiomer cannot be given by glycine, except you remember it, except glycine. Every amino acid are the enantiomer. These all non-polar amino acids are unique in nature because they are giving the hydrophobic effect. Hydrophobic effect. So what is the meaning of the hydrophobic effect? It whenever it will present in the protein, it will be very centered in this protein. So it will not have any contact with the water outside. So outside water will be contacted with the only polar, but these are the non-polar amino acid which is will be in the center, and it is stabilizing the protein with the hydrophobic effect. Okay, so it is stabilizing the proteins with the hydrophobic effect. Now, if you will ask about the glycine have a hydrophobic effect, but it cannot stabilize the protein for because of its small size. So glycine is a small, and it cannot not contribute to hydrophobic effect. Okay, it will not contribute to the hydrophobic effect. So most of the time, sometimes they can ask whether the which of the amino acid will help in the stabilizing of the protein. So not glycine will not contribute for the contribute for hydrophobic effect. Phobic effect. Hope so you clear. Hydrophobic effect, we just willing to know about the stabilization for protein stability. Among this all, you will see this is the sulfur containing amino acid methionine, but methionine cannot help in the protein folding. Okay, it will not help in the protein folding. We need sulfur bond. We need sulfur covalent bonds for make a folding between the amino acid. So it will complete help in the folding of the protein. But this sulfur is not free. This is a not free. Okay, this sulfur is not free. It from one side it is bound with the C2H4, then another side it is bound with the CH3. So it is not free. That's why it's unable to not give. I should write not give protein folding. Okay, not help in protein folding. This is all about the non-polar aliphatic amino group. So in every amino acid, what you notice, there will be amino group in this. Yes, here also amino group, here also amino group, here also, here. In every amino acid, if you see the amino group will be there. But what it is, what you found here, is this any amino or another group? This is NH2 plus. Other are the NH3. So this is not a amino group. This is a amino group. So you note it down. Proline. You note it down. Proline have amino group. Amino group, which make it cyclic, okay, which make it cyclic. So what if the proline is cyclic? Will it affect any protein? So because of it being the cyclic, the proline reduce the structural flexibility of protein. It will reduce structural flexibility. It will reduce structural flexibility of protein. Got it. So here we cover the non-polar amino group. Okay, here it is written aliphatic R group. But if I will ask what are the non-polar amino acid, so you will add in non-polar amino acid you will add the tryptophan as well as the tyrosine. Sorry, tryptophan and the phenylalanine. So you will add, you just add on plus. This is what tyrosine. We need to add tryptophan and phenylalanine and tryptophan. So you have to add phenylalanine and tryptophan in the non-polar amino acid, but not aliphatic. These are the aromatic. We'll see now. So tryptophan and this both are non-polar, and tyrosine is the polar amino acid. So what do you mean by the polar and non-polar? As you know, polar will not going to react with the water and polar can react with the water. Now, why it is called as an aromatic? Because it have a cyclic ring. Whenever you will see this cyclic ring, these are the consecutive double bonds will be there. Okay, so double bond, single bond, double bond, single bond. It can go for the resonance again and again and again. So whenever the light will hit to this, whenever whenever the light will hit this, then it will absorb this light, and after absorption it will go for the resonance. So aromatic compound is the very important thing. These are the why it is important, because why it is important, aromatic compound amino acid absorb light. Okay, absorb, wait. So as you can see, aromatic compound can absorb the light, specifically UV rays. Specifically what? UV rays. So what we are doing, we are take how we can absorb. We can absorb their reading in the spectroscopy. Spectroscopy. So in case of spectroscopy, in a spectroscopy, I think this color is visible or not. I will change this color. I think it's not visible. So spectroscopy will contain one CUET. Okay, it contain one CUET in which you will put your sample, in which you will keep your sample. So this is the sample you kept in this. Okay, the sample which you kept here, now the rays will be passed. So there will be, suppose, eye intensity of the rays are passed, but only two rays are coming. So this will be the intensity I node. So some of the intensity of the light is absorbed in this cuvette now. So how much absorbance happened in this? So what we can do? So absorbance will be equal to the log of emittance of light intensity and incident intensity. Okay, so this is the incidence intensity and the emittance intensity. What we studied here, this is the related to the Lambert's Lambert Beer's law. So in this Lambert's Beer's law, the sum of the light will be absorbed by the this solution. So it depends upon the how much concentration of this solution is there also na. So in accord, if we'll see according to the concentration, the absorbance will be equal to epsilon C into L. So what is this? It is a molar extinction coefficient. What it is? It is a molar extinction coefficient. Its value will be like mole per centimeter. Mole per centimeter. Its value is mole per centimeter. And this is a concentration. Concentration. Its value will be, its unit will be what? Gram per ml. Gram per ml. And this is what? This is the length. This is which length? It is the length of this CUET. How much length it is? So length is in the decimeter. You can write decimeter. You will write, or you can take as a centimeter. So this is all about the Beer's Lambert's Beer's law. This is the very important concept because in the examination they will ask this type of questions on the Lambert's Beer's law as well. And you will see this tryptophan will have the indole group. What it is? This is the indole group. Okay, this is the indole group, and this is the benzene, the methane, and then one more is there. Hydroxyl group will be there in the tyrosine. Where it is? This. So because of this, also it will become little polar. Hydroxyl group will be there now, free hydroxyl, which can contribute into the polar, making the amino acid more polar. Now we'll see the polar uncharged. So polar uncharged, in this you will find the hydroxyl group of the polarity like serine, amide group, sulfhydryl group of this. So this is the important thing. This is the very important thing. In cysteine you will get the sulfur. So there are two amino acids which have the sulfur. One is the cysteine, another is the methionine. But this cysteine will help in or participate in protein folding. Okay, protein folding. This will only help in the protein folding. Another will not going to help in the protein folding. Threonine, serine, and these are little polar also. Like this have the hydroxyl group. This also have the hydroxyl group. This have the NH2 group. So this will have the NH2 group. So asparagine and glutamine. What is the name? This is a later of their name. Asparagine and glutamine Q. So they are polar uncharged. Means they can become the polar if the pH will change. But it is in normal pH 7, they are neutral. What are neutral? The polar uncharged amino acid. Uncharged amino acid. Now, now we'll move the polar charged amino acid. So polar charged amino acid can be of two type. Either it can be positively charged. These are the polar charged amino acid. So come to this point again. This is a tyrosine is a polar uncharged. Coming back, polar charged amino acid are the either it can be of positive charge, either it can be of negative charge. So if you'll see the positively charged amino acid are the lysine, lysine, arginine, and histidine. So lysine, arginine, and histidine are the polar charged amino acid. They have the positively charged. Usually they will have the positively charged. Like this, extra they have. You will ask, sir, there are also negative charges there. There are also negative charges there. There also. So how you are saying positively charged? So this negative will be counter with this positive, this positive. So it will be neutral. But in net charge, if you'll see, net charge they will have the this positive charge, and another in this imidazole ring. Okay, this is the imidazole ring. So this is helping in the resins also. It will help in the resin and the chromatography techniques. It will help chromatography techniques. It will used. Okay, used in chromatography techniques because it will bind with the resins and it will bind to the another amino acids. So now negatively charged is the aspartic acid and glutamate. Aspartic acid and the glutamate have the carboxyl group, free carboxyl group, which will help in the negatively charged. Arginine have the free amino acid like guanidinium, and histidine have the imidazole, and lysine will have the small free amino group. There are two more amino acid which we studied about. The 21st amino acid. Okay, 21st amino acid is what? Selenocysteine. So as you know, this is a selenocysteine means it is derived from the cysteine only, but it have a selenium instead of it have a selenium replaced what? Selenium replaced what? Sulfur. Selenium replaced sulfur. Got it? So this is the amino acid which is coded by the one stop codon also, that is called UAG. Lu aa gaya. Okay, you remembered it. So this is a Hindi word which we used to say Ullu aa gaya. My teacher taught me. So I remember from him. Ullu aa gaya means what? Owl came. So UAG code for the selenocysteine. Got it? And it is also a stop codon. Stop codon. Even it code for 21st amino acid. Selenocysteine. So it will also present in the this selenocysteine is present in the selenocysteine will present in the glutathione peroxidase. Okay, glutathione peroxidase, as well as it is also present in the format dehydrogenase. Format dehydrogenase. Format dehydrogenase. Second is the 22nd amino acid. In this 22nd amino acid, if you'll see, this is the lysine modification, not modification, sorry, pyrolysine, similar to the lysine. So pyrolysine is coded by the sorry, I did some mistake. You should write UGA, Ullu Gaya. UGA. And this is coded by the UAG. Important thing about the pyrolysine is found only in found found only in the archaea. Archaea, like some bacteria, it will found only in the some bacterias, archaebacteria. So if I will ask you what name, what unit letter you will give for the naming, what single letter you will give with the selenocysteine? Can you find it any name? Serine already given, okay, S already given. So what name you will take for selenocysteine? They used to give the U. What they give the letter? They gave letter U. I cannot guess this U. I don't know how, but they given like this. For pyrrolysine, they given the O as a name. So this is the graph which is taken for the absorbance. If you'll see, the most of the absorbance is happened in this range around. Okay, so around 272, the line is not straight even. So you can write the most of the absorbance, absorbance for protein. We used to take absorbance for protein 280 nanometer. We used to take the absorbance for the 280 nanometer, but you will see the phenylalanine. Phenylalanine is not a good absorbent for the light, so their graph is very less. But you will see the tyrosine is okay, and tryptophan is the best for absorbance. So what do you think if you will apply the UV rays on the amino acid and the proteins? Then will this protein is having any damage from the UV rays? Yes or no? So if it is having any damage, then why we are using the UV rays? Think and tell me. In your comments you can tell, or you can tell me the U biology app as well. So this is all about the uh, basics of the amino acid, but you mention it here. D form of the amino acid. D form of amino acid is 99% in protein. So D form of the amino acid is more popular, like more acceptable in the proteins, but L form amino acid is very less in amino acids. So hope so you understood these things. We'll see some other amino acids which are uncommon, but these are the found in the amino acid. So these are not the modification, just uncommon amino acid we are going to write. Uncommon amino acid. Okay, this will be the modification only. We'll see. So what is that? Gamma carboxy glutarate. Okay, carboxy glutamate. So gamma carboxyglutamate, if you'll see, it will present in the prothrombin. Where it will present? It will present in the prothrombin. It's present in the prothrombin. What is the role of the prothrombin? So the role of the prothrombin, it will help in the, actually it convert into thrombin, and then it will help in the blood clotting. So it will bind with the calcium, and then it will convert it into thrombin, and then it will help in the clotting. So gamma carboxyglutamate also present in the calcium binding proteins. Binding calcium binding protein. So this was the first one. Another is the 4-hydroxyproline. 4-hydroxyproline. So this is the 4-hydroxyproline is what? The 4-hydroxyproline will present in the fibrous fibrous collagen, or you can say collagen fiber, it will present. Now we'll see desmosine. Desmo. So desmosine is what? Desmosine is the derivative of lysine only. Okay, it is a derivative of lysine, and it will present or it will help in the formation of fibrous elastin protein. Fibrous elastin protein. If you want to give me any suggestion regarding the color of the pen or background of the page, if it is visible or any changes is required, you can tell me in the chat box as well. So there are different type of modification will be occur in the amino acid, like phosphorylation, adenylation, acetylation, and these are helping in the formation of the different protein for the different purposes. We cannot define any even a single, like phosphotyrosine will be there, adenyl tyrosine will be there. So for the other purposes, for the different functioning, it will be present in it will modify accordingly. Now we'll see the non-standard amino acid. Non-standard amino acid. So there are so many non-standard amino acid, but I want to tell you the most important is the ornithine. Ornithine and citrulline. So these are the intermediate. These are the intermediate for the arginine synthesis. Intermediate of arginine and urea of and urea. Okay, students. Actually, it is taking a lot of time to complete this lecture, so I will split this lecture in the two part. The another part will be there uh, here on your right side corner. There will be one link for this another part. You can tap on that and you can go for the Henderson-Hasselbalch equation and the amino acid isoelectric point and deuterions we'll read in the another lecture. So thanks for watching till now. We'll meet in the next video. Jai Hind.